Hello, my lovely anatomists and physiologists. Michele Glass here. We left off with a discussion of how red blood cells are made, erythropoiesis, and we talked about this process being coordinated by the hormone erythropoietin, which remember is being produced by the kidney and is released when our blood pressure is low or when our oxygen levels are low to make more red blood cells. And we said that this process is occurring in the red bone marrow, and that we're starting from this hemocytoblast, myeloid stem cell. We go through a progression of different developmental changes until we finally get this mature erythrocyte or red blood cell. And so that's our topic for today. What does that mature red blood cell look like? Okay. And so our mature red blood cell, we're going to see, is going to be 7 to 8 micrometers in diameter. And this is going to be super important because some of our blood vessels, our capillaries, can be as small as 4 micrometers in diameter. So our erythrocytes have to be flexible and be able to like bend and squeeze into these vessels that are half as large as what they are. And so this is the real reason why that reticulocyte has kicked out nucleus, endoplasmic reticulum, Golgi apparatus, mitochondria. This is why we're kicking out all those organelles. So now we have this flexible, bendy cell that can squeeze through these super tight spaces. Now, because we've kicked out all of those nuclei and organelles, I said all of those nuclei as if one red blood cell really only has one nucleus, but because it's kicked that out, it can't repair itself. And so we'll see that your red blood cell is going to be in circulation for approximately 120 days until it's beginning to really experience a lot of stress from traveling through all of these tight spaces, and it kind of loses its ability to really move through the blood vessels. It loses some of its flexibility. It's sort of, I like to think of it as sort of becoming stingy with the oxygen. So it doesn't let go of the oxygen the way that it should. And so it begins to really not function the way that it should. And so we'll see that after about 120 days, we will actually be removing these old, damaged, worn out red blood cells. And we'll be talking about that process in just a little bit. So we've mentioned already that there are few organelles left in the cells. So that's kind of how we were getting to this flexibility. And that was really important in terms of why we have this very precise lifespan. Because of this, we also see that these cells are limited to the process of glycolysis, which is where you take a glucose and then anaerobically break it down into ATP and basically pyruvic acid. And so ATP is, of course, like our energy molecule for the cell. And the key feature here of the anaerobic respiration is and glycolysis is that like the red blood cell we've talked about really prefers glucose to make its ATP. And this is why it doesn't have a mitochondria, right? It doesn't have a mitochondria anymore. And so it's not able to do aerobic respiration. We mentioned like it can't really repair. So let's list that here. We'll see that it has a shape of what's called a biconcave disc. And here, I want you to picture a Smartie candy. A Smartie candy. So it's a disc shape, it's round, and it's indented on both sides. That's your biconcave disc. Okay, so that's your shape, but instead of that hard candy, it's that flexible cell. And then we describe them as being able to form what are called rouleaux. And this is talking about like a stack of coins or a roll of coins. And so this is like, as they move through these super tiny spaces, of the capillary, they are able to do it in a nice, precise line. Yeah. So that they're able to squeeze through and move through in a nice, orderly fashion. And so this is enabling these cells to go through a 4 micrometer diameter capillary when they're twice as big as that. And then I think the other thing that we'll mention here is that this is a cell that's full of the molecule called hemoglobin. And it's the hemoglobin that's going to actually bind to and transport oxygen gas and CO2 gas. And so really your erythrocyte is just this package of this hemoglobin molecule in order to do this transportation. And what we'll see is that a single erythrocyte can have, let's see, 300 million hemoglobin molecules. Yeah. So it's basically just this biconcave disc, flexible, full of hemoglobin. It's maintaining those enzymes to do glycolysis so that it can keep moving through the body for about 120 days, right? And so let's talk next about that hemoglobin molecule. And so I have over here a photo that's taken from Creative Commons license on Google. So you can do a quick search for yourself as well. Hemoglobin is a protein. And so the protein is composed of 4 subunits. And subunits, remember, we're talking about a primary structure of the protein, which is a chain of amino acids, a secondary structure of the protein, which is folded up either into what we call an alpha helix or beta pleated sheet. And then the tertiary folding is going to be that hydrogen bonding between R groups, right? And so each one of these 4 subunits or chains has its own primary, secondary, and tertiary folding. And then this is the quaternary folding or complex where we see these 4 subunits coming together. And so we just describe them as having 2 alpha chains and 2 beta chains. And for us, that's not super important, but if you are reading about sickle cell anemia, then those different subunit chains will be mentioned. Now notice at the center of each one of these chains. So just imagine you have basically these 4 kind of protein globs. And then at the center of each sort of protein glob, you have a heme group, which on these diagrams looks like a disc. And at the center of that disc, you have an iron ion. So iron is a cation, okay? And so you have 4 subunits, which means you have 4 heme groups, which means you have 4 irons. And what we'll see is that it's the iron that actually binds to the oxygen. The iron at the center of each heme group actually binds to the oxygen. So what this means is a hemoglobin molecule can bind 4 oxygen molecules because each subunit has a heme and each heme has an iron. So that means a single hemoglobin molecule has 4 oxygen binding sites. It has 4 heme groups, 4 irons. That also means that iron is going to be an important component to making red blood cells. Now remember that a single cell, so one red blood cell, has about 300 million hemoglobins. And we can abbreviate hemoglobin with a capital H lowercase b. There's several different abbreviations, but we can use that one in our materials. And so if a single red blood cell has 300 million hemoglobin molecules, and each one of those hemoglobin molecules can transport 4 oxygen molecules, a single red blood cell can transport 1.2, I'm doing the math for you, thank goodness, right? 1.2 billion oxygen molecules. So that's quite a lot here. When we talk about hemoglobin saturation, and we're going to talk more about this in the future, what we're really saying is how many of these seats is occupied by oxygen. So if you want to think of like the iron as a seat on the boat that transports the oxygen, right? That means like each boat of hemoglobin can hold 4 oxygen. And so if we talk about hemoglobin saturation as being like 100%, that means like all the seats are taken. So that means oxygen is binding to all 4 of those particular heme groups. Now, if we experience what's called hypoxemia, then what we have going on here is a low blood oxygen, meaning we don't have all those seats taken. And what we're going to see is that the kidney is going to increase the production of EPO in order to increase our number of red blood cells. And so by giving us more of these boats, more of these oxygen transporters, hopefully we can get that oxygen to our tissues. As we were talking about this, and I mentioned that the iron is this critical component of the hemoglobin that's actually binding to the oxygen, that does remind me that we need to add a few details to our discussion of erythropoiesis. So go back into your notes here and let's make sure to add just a few details. So when we talk about this production of erythropoiesis, we said, okay, it's dependent on this hormone erythropoietin, but then we also see that we need to have certain trace elements. Trace elements mean that these are things that you need to get from your diet in very, very small quantities. And this includes iron, which we just mentioned is part of that hemoglobin molecule. It's actually the thing that's binding to the oxygen. We'll see that we need copper, we'll need zinc, and then we'll need B vitamins here to help really stimulate this production of red blood cells. Okay. Up next, we're going to talk about what happens when we get to that 120 days and our cells are old and tired, and how does the body process that old and tired cell? So stay tuned, and as always, take care of yourselves and each other.